Es un honor para mí estar aquí como moderador de esta sesión, que es la segunda sesión de alimentación de la yegua gestante. Tengo el honor de presentar a la doctora Morgan Robles con el tema alimentación, ambiente materno y fenotipo. Ella es un miembro que se encuentra en el National Institute of Scientific Research en Laval, Canadá. Su trabajo de investigación son los efectos del estrés materno, la ansiedad y la depresión en la función placentaria y el desarrollo de la descendencia en humanos. Su formación incluye una maestría en fisiología del ganado, en Agrocampus, en la Universidad de Rennes, en Francia. Su doctorado en el Instituto Nacional de Investigación Agronómica en Francia, con el trabajo de investigación Efectos de la nutrición materna y el metabolismo durante el embarazo en la función placentaria y la salud de los potros, con interés primordial en los efectos de la paridad materna, la ingestión de almidón y aminoácidos y la obesidad en la estructura y función placentaria, y el crecimiento en los potros, el metabolismo de carbohidratos, lípidos y el estado osteoarticular. Sus estudios de postdoctorado son los efectos de la transferencia de embriones y la paridad materna en el desempeño de salto. Su investigación se basa en experiencia y investigación en el desarrollo de Reverdi Nutrition Equine en Francia, el desarrollo de una nueva granja experimental y en la comunicación científica. Tiene 5 publicaciones en revistas científicas de alto impacto. Vamos a tener una serie de preguntas y respuestas después de las pláticas de los doctores Robles, Quijano y García Pasquel. Entonces es importante contemplar que las preguntas que sean en inglés para la doctora Robles la vamos a poder observar y nosotros vamos a hacer la traducción correspondiente, y para mí es un honor presentar a la doctora Morgan Robles. Muchas gracias.
Good morning, everyone. Well, good afternoon, actually. I hope everyone hears me well. So thank you for the introduction, and thank you also for inviting me to present these 2 presentations, so the one today and another one tomorrow. So today I will talk about feeding, maternal environment, and phenotype. I will not really talk about horses today because I will more present what is the role of the maternal environment on the development of the offspring in general. And tomorrow will be more focused, like only focused on horses actually. So I will first talk about a history of what is developmental origin of health and disease, because it's important to understand where we come from and what we learn through the centuries. Then I will define the concept through examples. And we will talk a little bit about mechanism. So it's a molecular mechanism. It can be a little bit complicated at first, but it's very important to understand why we observe what we observe, what happens. And we'll also talk a little bit about the placenta. And then I will present some examples in mainly other species, because you will see tomorrow that we don't know a lot of things in horses. So we'll present some example in other species to show you how the maternal environment, and especially the nutrition, can affect different organ development in the horse. So first, history of what we call either developmental programming, fetal programming, or developmental origins of health and disease, what we talk about DOHAD. So it's been thousands of years that the physicians have understood that there was a link between the maternal environment and the development of the fetus. If we go as far as for Hippocrates, for example, so before like 500 years, 400 years before Jesus Christ, Hippocrates said that now it is just the same way that the child in the womb lives from its mother, and it is on the condition of the health of the mother that the condition of the health of the child depends. So there was already a link between the health of the mother and the health of the child that was understood at that time. Later on, if we go further with Leonardo da Vinci, you can see that he said that one will, one supreme desire, one fear that the mother has, or mental pain, has more power over the child than over the mother, since frequently the child loses its life thereby. So it was like there was an understanding that the maternal psychology affected the development of the child and could even lead to abortion or stillbirth. A few years after, another physician, Paracelsus, said an infant in the mother's womb is as much in the hands and under the will of the mother as clay in the hands of the potter, who from it makes whatever pleases him. So it's really the idea that what happens to the mother will be impressed upon the fetus and will affect the development of the fetus. So there was this idea of what happens to the mom will happen to the child and will change its development. But we have to wait until 1934 to see the first paper, the first scientific publication that really shows the correlation between the maternal, like the early environment, and the health of the adult later on. And this paper that has been published in 1934 shows that there is a correlation between the constitution acquired during the first 15 years of life and the risk of the death rates of the adolescents and the adults. So basically what they say is that if a child has a poor starting of life, so a poor environment, poor nutrition, poor health, during its first 15 years of life, it will have a higher risk of death when he's an adolescent or a young adult. Then we have to wait until the 70s to really see the first studies that really continue to show those correlations. For example, here, 1977, the authors showed that the infant mortality in very, very early life was positively correlated to the death of cardiovascular disease at adulthood. So children that had the places where they had the high infant mortality, so where children had poor nutrition, poor health, etc. during their beginning of life, when they became adult, they have an increased risk of having heart diseases later on. And then we arrive in the 80s with the studies of the Professor Barker. His team did a lot of epidemiological studies that really showed that there is really a correlation between the mortality in early age of the children and the risk of developing cardiovascular disease when they are adults. And they continued more in details their work, and they showed that it's not only the poor environment that affects the risk of developing disease later on. It's also the weight of the child at birth or at 1 year of age. And what you can see is that if you have children that are born with a lower weight, or that still have this lower weight at 1 year of age, when they become an adult, they have more risk of having cardiovascular disease, diabetes, and what we call now the metabolic syndrome that they called Syndrome X at that time. So this epidemiological studies that were like this led to what was called Barker's hypothesis, which has been it was not very well accepted at first. Now we did a lot of other epidemiological studies, a lot of experimental studies in animals, and what was an hypothesis at that point became something that is now validated, and we understand it quite well what's happening, and now it's called developmental origins of health and disease instead of Barker's hypothesis. So now I will define the concept through some examples. So the main example that I will take is the Dutch famine of winter 44-45. So during the Second World War, so during this winter, there was a German blockade of the Netherlands. So the people in the Netherlands stopped receiving food, and there was a famine because of that. So the mean caloric intake of the people was 400 kilocalories per day. For a pregnant woman, so in humans, the energy needs are up to 2,500 kilocalories per day. It means that the women that were pregnant during this famine received only 16% of their needs in energy, or 6 times less. So these women were pregnant either in early pregnancy, middle pregnancy, or late pregnancy, because the winter only lasted for a few months. So the children were affected at different times of exposure during their development. Moreover, what is interesting in this cohort is that after the end of the war, the blockade was alleviated, and the children that were born at that moment, they did not have to encounter any famine anymore. So they were normally fed afterwards, and more in a country with a lot of food than not enough food. So their postnatal environment was very, very, very different from their in utero environment. So from a lot of studies have been been performed on this cohort. So I will present you the main results that they saw based on the different time of exposures during the pregnancy. So when the children were exposed in late pregnancy, when they were born, they had a lower weight at birth and they were smaller. And they continued to study the people until adulthood, and they saw a decreased glucose tolerance in those people. When the children were exposed at mid-pregnancy, they were also lighter and smaller at birth. And when they studied the adulthood health, they saw that they had a decreased glucose tolerance, which can lead to diabetes at some point, some kidney problems, and an increased risk of having airway obstruction disease, such as asthma. And when the children were exposed in early pregnancy, you can see that they were heavier and bigger at birth compared to the other kids. And in adulthood, they observed that they had a higher amount of cardiovascular disease, some lipid profile problems, and obesity, which kind of looks like the metabolic syndrome. After, they also studied if the children were exposed after birth. So some were exposed from birth to 14 years of age. And you can see that also based on the age that they had when there was the famine, you see that the effects are also different. Like from birth to 1 year of age, they did not observe any effect. From 1 to 5 years of age, when they studied the health of the individual at adulthood, they see that women only had an increased risk of having type 2 diabetes. The ones that were exposed from 6 to 10 years of age, no effect either. And the ones that were exposed just before starting of the adolescence had an increased cardiovascular diseases and type 2 diabetes. So what this very big study tells us? It tells us that first, the early environment of an individual will influence its long-term health, and especially its risk of developing non-communicable chronic diseases such as diabetes, cardiovascular diseases, lung problems, etc. Secondly, the period of development affects this effect, because you know that you've seen that the effects are different based on when the children have been exposed, if it was in early pregnancy, in late pregnancy, or during their childhood. So the period of development will have different effects on their development. And third, there is a difference between the males and the females. It's not very obvious in this study. I will show you more, some studies more, but there is a sex-specific difference, and males and females don't have the same effects. So let's see more example on sex, what we call sex dimorphism. So here so as I said, I will mainly present studies on other animals than horses, because we don't know a lot in horses, and there have been a lot of studies that have been performed in other animals. I will try my best to present herbivorous animals, but sometimes nothing exists in these animals either. So here we have rabbits. So the rabbit does have been fed a high-fat diet or a controlled diet from puberty to the end of gestation. They sacrificed the rabbit at 20 days of gestation, so 3 days before birth. And for the both sex, they observed that the babies were smaller and lighter, so there was a fetal growth restriction during the pregnancy in the high-fat diet group. But when they studied the lipid content in the fetal blood, they observed that only the males were affected by the maternal diet and not the females. As you can see here, you see an increase in different fatty acids in males only, but not in the blood of females. So they studied the placental lipid storage to understand if the placenta could explain this difference between the sexes. And what they saw is that for the babies that were born from the high-fat diet, those, you can see a higher, like an increase in lipid storage in the placenta. So the placenta will try to adapt and will try to protect the baby by storing the fat. So you can see here the white dots or the lipid droplets, or here it's the red dots, it's the lipid droplets. So here it's the high fat group, and here it's the normal group, the control group. And when they studied the content of lipids in the placenta, you can see that it's mostly in females' placenta that you see a difference in lipid storage content. So it looks like males are more affected because the placental adaptations were more, there were more placental adaptations to protect the females. So there is really differences in sex between sexes because of the genetics, but also because the placenta can adapt. If we come back to the Dutch famine, the authors also showed that when the children that were no adults, so that were undernourished in utero, when they have their own children that were not affected by the famine at all, their own children were born in the 70s, 80s, so they were not affected by the famine at all. When they had themselves children, their own children had a decreased birth weight. So it's like the effects that are observed in the individual can be transmitted through the generations. And there is this study on rabbits. So it's not really nutrition per se, but it's rabbits that have been exposed to diesel exhaust during their pregnancy. And it's a good model of nutrition, fetal undernutrition, because the placental blood flow and vascularization was decreased. So the fetus were growth restricted, and it shows that they were undernourished during the pregnancy. When they studied the first generation of those rabbits at adulthood, they saw a sex dimorphism between males and females, with males having hypertension, hyperglycemia, an increased fat weight. While for the females, it was more the bone weight and density that was affected. When they mated the females with a male that was not affected by diazyl exhaust, and those females were only affected in utero. After being born, they were normally raised like normal rabbits, no more diazyl exhaust. So when they mated those females with a normal male, they studied the second generation, and they see some alteration in the placental development and some changes in the lipid metabolism in the fetus. So they see that there was a transgenerational transmission from the grandmas. So what are the developmental origins of health and disease? What does it mean? So as I said earlier, it means that the early environment of an individual will influence its long-term health. The periods of development affect those effects, as I showed, and there is a difference between males and females. But also, this can affect more than one generation. And even if the first generation is not affected by, like, that does not encounter this environment, deleterious environment, it will still be affected. So it will be transmitted through generations. So the concept of DOHAD, like, there was a society that have been created, it's called the DOHAD Society. They have congresses every 2 years, they have a journal. Now it's really something that has been validated and confirmed, and we really understand better what's going on. So try to conceptualize a little bit what's going on. So when you are an embryo or a very young child, you have a very high plasticity. It means that you're able to adapt to your environment. But as you grow older, your ability to adapt to your environment will decrease. On the contrary, the detrimental effects of your environment will increase. So your risk of developing long-term chronic non-communicable disease such as diabetes, cardiovascular disease will increase with your age. This is what we observe normally in the population. If you are in a deleterious environment, so as I showed you, famine, for example, during your early life, your risk of having diseases will increase. And it will increase furthermore as your postnatal environment, so the environment that you will have at childhood or adulthood, will not match the environment that you had when you were either embryo fetus or a baby. It's exactly what happened for the Dutch famine study, because the individuals were undernourished during their in utero life, and then when they grew up in a society that had plenty of food, so they were not adapted to this different type of society. On the contrary, if you want to decrease the risk, you have to have an intervention as early as possible, because the earlier the intervention, so the earlier the change in nutrition, or for example, if you have an obese mare, you just try to decrease the amount of fat that the mare has, that she's not obese anymore. The earlier the intervention, the more the more important you will decrease the risk for the individual to have disease later on. So you can see here, for example, if you have an intervention very early, it will decrease a lot. If you have an intervention later during childhood, for example, it will decrease less. And if you try any intervention at adulthood, the effects will be very, very, very small. So still in the conceptualization of everything, you have an individual. This individual has what we call a genotype, or a genetic information, genetic patrimony. This individual will be in a prenatal environment. That is what it is. this individual will adapt because he has a very important plasticity when he's very young to survive in short term. So that's what we call the immediate and predictive adaptive response. You adapt to survive in short term. So if, for example, you are undernourished in utero life, you will adapt in order to be able to survive in a world that does not provide a lot of food, a lot of energy. That will lead to what we call the thrifty phenotype, basically individuals that are able to store a lot of energy. But if the postnatal environment doesn't match the prenatal environment, you will have more risk of developing non-communicable disease because you are not able to respond adequately to this environment. For example, the individual that is very, very adapted to an environment that has a very poor nutrient and energy content, and then that will arrive in the United States, for example. This individual is not adapted and will have problems such as obesity, diabetes, etc. This is what we call mismatch or second hit. So now I will talk a little bit about the mechanism and especially the molecular mechanism. I will try to be as clear as possible because it's complicated even for the scientists, but it's very important to understand what's going on. And I will talk a little bit about the placenta. So first, everyone knows that a clone does not look like its donor. So I will show you some examples and then I will explain you why. So first example is copycarbon or copycat. It's a cat that has been cloned in Texas A&M University. So here is the donor, here is the clone. So you can see that the donor and the clone don't have the same color pattern in their hair. Here is, so in my PhD laboratory, we had clones of cows. So here is the donor, and here are all the clones. So they do not have the same, they are not the same age, but mostly if you look at the back of the cows, you can see that the pattern of the white and black spots are different between all the cows. They do not really look like they are the same individual. Same here, if we focus on horses, you have Top Gun, and you have its clone. And you can see that the socks are different between the clone and the donor. Also, the white thing here and the skin on the nose is also different between the two animals. But they share exactly the same genetic information. So what is different between a clone and its donor, and why a clone is different from its donor? First, it's about the mitochondrial DNA. I will not really talk about that, but it's true that the clone does not have the same mitochondrial DNA than the donor, but it doesn't explain everything. The most important thing is that, first, the clone has been produced in a petri dish. It has been fed with synthetic medium that you feed the cells, while the donor has been produced in a normal cat or a normal animal, has been fed the nutrients from the mother directly. So it's the first difference between a clone and its donor. The second one is the maternal environment. Here you have copycarbon with his surrogate mother. That is not the same mother as his donor. So the whole embryo and fetal development did not occur in the same place with the same environment. So you can see how the environment can affect even the hair, the pattern of the hair of the animal. And this is because of epigenetics. So epigenetics comes from epi, which means above in Greek, and genetics or genome, which is the genetic information. It encompasses all the molecular mechanisms that regulate genetic expression without changes in DNA sequence. So you don't have changes in DNA sequence, but you have changes in genetic expression. The way it's a little bit like you have one book. you either read the even chapters or the uneven chapters. Even if it's the same book, the stories will be different. It's exactly that. So the epigenetic marks are heritable. They are heritable in your own body. So between your cells, when your cells, they duplicate and reproduce, they will give their epigenetic marks to the other cells. But also your gametes will also sometimes be transferred to the other gametes, such as spermatozoids or ovocytes. It's also reversible, so it can be reversed until some point. As I showed you, there is a high plasticity in early life, and this plasticity decreases as as you age. And those marks are affected by the environment. And this is what Conrad Waddington explained very well with his epigenetic landscape. It's why your lung, pancreas, and intestine cells, even though they share the exact same genetic information, they do not look like, they have a different structure, they have a different function. So the epigenetic marks, we have approximately 3 of them, like 3 big groups: the DNA methylation, the histone modifications, and the non-coding RNA. So I will come back more into details to what are these mechanisms. So first, to be sure that everyone follows me, so the DNA, which holds the genetic informations, is under the form of a double helix. Because it's a very, 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 very long molecule, it needs to be compacted into a chromosome to fit into the cells, actually. For the compaction, it will wound around the nucleosomes. And what we call nucleosomes, which look like the thing where you put this, when you sew, it looks like that. And the nucleosomes are made of proteins that are called histones. And for what we call the transcriptional machinery, so it's all the proteins that will be able to read the genetic information. For those machines, so for this machinery to be able to read the genetic information, the nucleosomes need to be far from each other. So what we call the chromatin need to be opened. If you close the chromatin, if you get the nucleosomes to be closer to each other, the expression will be like the machinery can't access, and there will be no expression, so the genetic information will not be read. This is basically what are the epigenetic marks and how they work. For example, the DNA methylation, it's the adding of a methyl group, so CH3, on a cytosine. Cytosine is the base of DNA, so it's it's the alphabet of the DNA that holds the genetic information. So the cytosine will be methylated, but only in what we call a CpG island. So you have a cytosine and a guanine, another base. And when so this methylation, it occurs through the DNA methyltransferase. I will not enter into details, but you have to know that there are a lot of DNMTs, and they have different roles. Some remove the methyl group, some will maintain the methylation, so they have a lot of different groups. Usually, when you have a methylation, it will repress the gene expression, so the nucleosome will get closer to each other, but it's not always true. Then you have the histone modifications. So histone modifications, it's either acetylation, so adding of an acetyl group, or demethylation, or acetylation and methylation. So either you remove the acetyl or methyl groups, or you add the acetyl and methyl groups to the histones, so the proteins that are that build the nucleosomes. Usually, when you remove those groups, you rapproche the nucleosomes. When you add them, you make them be further from each other, and then you open the chromatin, and you can have a transcription. So it's a very, very complicated mechanisms. And for example, if you acetylate on a special amino acid, so lysine in histone, you have a decrease in the interaction between nucleosomes, so an active transcription. But if you add a methyl group to a lysine or an arginine, the effects will depend on what histone is methylated and the number of methyl groups that you add. So it's not black and white. It's a lot of different effects from, like, it's very, very complicated. But it's basically opening or closing the chromatin through molecular changes in either the DNA or the histones. You also have the non-coding RNA. Today, with the vaccines and the COVID, we talk a lot about messenger RNAs, which basically are the small molecules that will carry the genetic information between DNA and protein. Small RNAs are RNAs that do not code for protein. You will never have a protein from the small RNAs. They are small. And we have two different kinds. We have the microRNA and the siRNA. So the microRNAs are RNAs that can recognize a lot of different messenger RNAs. And when they recognize them, they will attach to them, and that will lead to a translational repression, because like the proteins that normally would recognize the messenger RNA and read the messenger RNA to transform it into a protein, they can't recognize it anymore. So there will be no protein from this messenger RNA, which will repress the translation. On the other part, you have the small interfering RNAs. Those ones are very, very specific. And same, they will recognize the messenger RNA, but they will lead to the cleavage and the degradation of this messenger RNA. So both small RNAs, small non-coding RNAs, lead to the protein will not be translated, and so it will lead to a decrease in the expression of the proteins. So how does nutrition can influence the epigenetic marks? So it's quite a complicated figure, but what I want to tell you is that we have what we call the methyl donor, which is the S-adenosyl methionine. From this S-adenosyl methionine, you will be able to either methylate the DNA or methylate the histones. This S-adenosyl methionine comes from the methionine, which is an amino acid that comes from the proteins from the diet. You can see that you can't have S-adenosyl methionine if you don't have vitamins B12, vitamins B9 or folate, vitamin B6, and a lot of different compounds that you find in the food also. So yes, the nutrition can affect these epigenetic marks because it can affect the methylation of the DNA. If we come back to the critical periods of development, as I showed you, that the effects are different based on the periods of development. You have to note that there is what we call the genome activation. So the embryo, until the genome activation, has a very, very compact chromatin. And then there is a genome activation, the chromatin decompacts, and everything will be red. Well, not everything, but a lot of things will be red so that the embryo will develop. And then you have all those periods that are very important and that are different. You have a whole period of organogenesis, the period of gonadal differentiations. Those periods are very, very important. And you should feed the right quality of food and the right amount of food to have a good organogenesis and gonadal differentiation. So all these different critical periods of development will lead to different effects because not the same organs or not the same, it will affect it exactly the same. Here is just to show you that the DNA methylation changes. So here you have the embryos, the zygote, the first step of the embryo, that has a very, very high DNA methylation. As I told you, the chromatin is completely compacted. And as the embryo will grow, the methylation will decrease and the chromatin will open, and the genes will be, the genetic information will be able to be read. And once you reach the blastocyst, so embryo, one embryo step, so it's the embryo that implants usually, you will see an increase in DNA methylation again. So you see that all those periods affect. So the chromatin is open or closed, so you can think that the environment will not affect the epigenetic marks the same way if the chromatin is already more opened or more closed. So here it's more in the mice, but it's quite the same in other animals. So just to again present you the critical periods, here is an example, some examples in the sheep. So if you have maternal overnutrition, either in preconception, early pregnancy, or mid to late pregnancy, you can see that the effects on the development of the lamb are different based on when an overnutrition was applied. You see an increased ovary weight if it was during periconception for the lambs, a decreased follicle numbers if it was in early pregnancy, and then changes in metabolism if it was from mid to late pregnancy. And when they studied the sheep at adulthood, you see an effect of the weight on the females if the mother were overfed during preconception, an increase in muscle fiber density if it's in early pregnancy, but a decrease in weight and a decrease, actually an increase in visceral fat over the subcutaneous fat if it's from mid to late pregnancy. Same for the undernutrition. You see different effects with different time of exposure. So it was true in human, it's true in the sheep, and it might also be true in the horse. So now just a little, very, very little part about the placenta. So the placenta is considered as the programming organ of the fetal development. It's because it's the organ that is at the interface between the mother and the fetus. It will adapt to what the mother gives as nutrients and everything to be able to protect and feed the fetus as better as possible. The placenta derives from the embryo. It's embryonic. It's not the mother that produces the placenta, it's only the embryo. So it has the exact same genetics and sex as the fetus, because the placenta has a sex. It is a transitory organ. It grows and develops only during pregnancy, and it is expelled at birth. So it's a good marker of what happened during the pregnancy. And it's an organ that is involved in fetal nutrition, fetal excretion, metabolism. In metabolism, it works a little bit like a liver. It's very good at detoxifying. It's involved in storage. It produces hormones. It's involved in gas transfer, and it has a huge role in immunology. So it's an organ that is often underlooked. It should be more studied. The placenta is able to sense the nutrient supply from the mother and to adapt in size, in morphology, to adapt its blood flow, to adapt its hormone production, and especially it produces a lot of growth factors. So it's a very, very important organ. And it is also affected differently if based on the periods of gestation. So here you see that different placental weight from sheep that were underfed during pregnancy. And you see that if the undernutrition is in early pregnancy, there is an increase in placental weight. But if it's in late pregnancy, there is a decrease in placental weight. So the placenta will adapt differently if the undernutrition is in early or late pregnancy. And we have a wide varieties of placentas, which means that we observe a wide variety of effects between species too. Just for example, here are the main categories, the main shapes of placentas. So here you have the horse, which is very close to the sheep, the pig, sorry. Here you have the ruminants, the carnivores and the primates, rodents and lagomorphs. And you can see that all the placentas have different shapes, but also different structures. So you can imagine that the effects will be different based on the species too. Just to have a very small focus on the horse, the equine placenta is diffuse and epitheliochorial. So what does it mean? Diffuse, it means that it covers the whole uterus. And epitheliochorial, it means that it does not invade the uterus. There is almost no blood when a mare gives birth. But it means that there are 6 layers of cells that separate the maternal and the fetal blood in the horse. So really, the maternal and fetal blood never come to, never share, never cross. But you can see that in the horse, the number of cells that separate both bloods is very important. And I will talk more about that tomorrow. So now, how the maternal environment would possibly affect the horse performances. What do we want in a horse? We want a horse that has a good muscle development and metabolism. We want a horse that has a good cardiovascular development, a good lung development, especially for race horses, for example, or eventing horses. We want a good bone development. We want a good brain development. We want a smart horse. I want a horse that is able to reproduce well. And we want a horse that has a long lifespan, that has longevity in his own sports or activity that they do. So today I will talk about all what is in white. The bone development, I will talk about it in horses tomorrow, and the brain development, there are not enough studies now to talk about that, but there is already things that show that maternal environment and especially maternal nutrition can affect the brain development. So first, muscle development and metabolism. So you have to know that the number of muscle fibers is set at birth. You are born with a set amount of muscle fibers, and that's it. If you go to the gym, your muscles will grow in size because the muscle fibers will grow in size, but you will not have more muscle fibers than the numbers you were born with. This is why in utero nutrition is very important for the development of the muscles, especially. So here, there was the study, the studies where ewes were either underfed or overfed during the gestation, or fed a control feed. At 19 days of gestation, so mid-gestation, in the sheep, they sacrificed the ewes and they studied the muscles in the fetuses, and you can see that the number of type 2 fibers, which has the fast muscle fibers, is reduced in the longissimus dorsi, so it's this muscle, in the babies of restricted or overnourished mothers. They also followed the babies until 3 months of age, and at 3 months of age, they observed that it was not only the number of type 2 fibers that was decreased, but also the area of the fibers, of the muscle fibers, was decreased in over and restricted groups in the semitendinous, so this muscle. And also that there was an alteration in muscle lipid storage in both groups, either an over lipid storage or an under lipid storage, which can mean that the muscle metabolism is not perfect, it does not work so well. So you can see that maternal nutrition, if it's not adequate, can affect the development of the muscle before but also after birth. And there is this study that has been performed in the rat that also shows that when the mothers were fed a high-fat diet, their babies had a decrease in proportion and size of the fast fibers, the fast type fibers. About the cardiovascular development, I wanted to talk a little bit about the horse because something has been done with that and it's very interesting. So in this study, the authors transferred the embryo of a thoroughbred in a Welsh pony, so it's a model of maternal fetal undernutrition, or the embryo of a Welsh pony in a thoroughbred, which is a model of fetal overnutrition. So here you see the thoroughbred that has been transferred into a Welsh pony that is significantly smaller than a normal thoroughbred. And here you see the Welsh embryo, so that is now a grown-up pony that has been transferred into a thoroughbred that is significantly bigger than the normal Welsh pony. And here are the same foals, so the thoroughbred transferred into a Welsh pony and the pony transferred into a thoroughbred at birth. So you can see that the thoroughbred that was transferred into a Welsh pony is dismature, has a very, very huge sign of dismaturity. They followed those foals later on, and they followed their blood pressure. So you can see that the ponies that have been transferred in the thoroughbred have an increased blood pressure. And they also followed how they regulated their blood pressure, and they show that in both groups, they have a not so efficient regulation of the blood pressure, which can lead to cardiovascular disease later on. So even if these models are very, 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 like, the effects are very important because it's more than just a small undernutrition, it's a huge under or overnutrition. You can see that even in the horse, changing the maternal environment can affect the cardiovascular development of the foals. Now for lung development, so as I showed you for the FAMINE study, the individuals that were exposed to undernutrition during mid-pregnancy had a higher risk of having asthma. Well, some people did a study in the rat. So they either fed a protein-restricted diet during gestation and lactation to the mothers or a controlled diet. And then they studied the function of the lungs of the offspring. And you can see that in females only and at 21 days of age, the offspring of the mares that had received the protein-restricted diet had a decreased compliance, which is the extent to which the lungs will expand, and an increased elastance, which is the pressure required to inflate the lungs. So basically, altered lung function in this offspring of protein-restricted mothers. Moreover, they also observed in both sexes that the resistance to the flow of air of the lungs was increased, and usually the lower the resistance, the better. So they showed that maternal nutrition could also affect the lung development, and I don't show it here, but they also show that, we have a kind of not a liquid, but something that is in the lungs and that will increase the transfer of oxygen from the air to the blood that covers the lungs. And they see that this surfactant also is altered. The composition of this surfactant is also altered in the offspring of protein-restricted mothers. So what about reproduction? I just show a short example because I will talk about reproduction more in the horse tomorrow. But here you see the goats that have been either not supplemented with selenium during gestation and lactation, or supplemented with the increasing amount of selenium. And their babies, the babies that received, from the mother that received the supplementation, they had increased weight of the testes, increased length, increased width, and also an increased density of the spermatogenic cells and the Leydig cells. So basically, testes that work better. And they also observed an increase in serum testosterone and testis testosterone in the offspring of the supplemented goats. So it also shows that maternal nutrition can also affect the reproduction of the offspring. Now for the lifespan, I like this study because it's very interesting. So it's a little bit complicated. You have mice that have been fed a normal protein diet or a low protein diet during pregnancy, or a normal protein diet during the whole pregnancy and a low protein diet during lactation. When their baby were weaned, they were either fed a normal food, CHO means control food, or a cafeteria food, which is a high-fat, high-sugar diet. And they studied the lifespan of the animals. So for the animals that have been fed either a normal protein diet during pregnancy or a low protein diet, the offspring of the low protein diet mothers, they had a decreased lifespan of 1/4 compared to the offspring of the normal protein diet. On the contrary, when it was during the lactation, you see an increase in the lifespan of the offspring. What is also interesting is that the high-fat, high-sugar diet decreased the lifespan of the individuals when they were fed this diet after weaning, but only for the mothers that were fed a normal protein diet or a low protein diet during pregnancy. But it did not affect the lifespan of the offspring when the mother was fed a low protein diet during lactation. So this study also shows that even just changing the amount of protein in the diet can really, really significantly affect the lifespan of the individual. So in summary, an inadequate supply of maternal nutrients can have profound effect on offspring development through several generations. And this effect can alter the athletic, the cognitive, the behavioral, and the reproductive ability of the individual. So feeding the mare is feeding the future foal, and along with genetics, early nutrition should be considered to improve and work with to improve the horse performance. So thank you for your attention.
Thank you so much, Dr. Morgan, for all your presentation. It was amazing and illustrated us quite a bit. We're going to have the question and answer period at the end of all the presentations. Thank you very much.